What is up, guys? Okay, so today I'm going to be giving you guys a bit of an insight into how I study for my anatomy lab practicals on the digestive system. Okay, so here's the first thing I'm going to do. I'm going to be watching a video that my study group put together on the small intestine. So this is going to be on the lower digestive system, small intestines, and urinary bladder. So this is the first video. The small intestine. Okay, that's the first video. So it was a bit more of the small intestines. You could see a bit of the mesenteries and the urinary bladder, and a bit of a division over there in the small intestines. And this is how we usually start off our slides with, usually just videos. Here's the next one. So it's going to be on the human digestive system. Let's check it out. A little bit. Parotid gland right there. Mandibular gland on the mandible. Urinary bladder. Okay, everything's all good with that. Okay, so here's the next video, 3D model of the digestive system. Or the parotid gland. duodenum. So a bit of a summary. So going from the mouth, down the esophagus into the stomach, and from the stomach it goes through the pyloric sphincter into the duodenum, through the jejunum ileum, and then into the colon or large intestine at the cecum. Ascending colon, transverse colon, descending colon, and the sigmoid colon, and then all the way down to the urinary bladder and out the rectum. That's a bit of a summary right there. Next one. So, one thing we digest a lot in my anatomy lab class is a mink or minks. And here's a digestive system video for it. We got a bit of the teeth labeled and some of its lower digestive system for you guys. And this one's for a cat, not a mink. Laryngopharynx. Ooh. Looks good. Next video, more into the cat digestive system. While they're playing music. Looks good. So, mesocolon, that holds, they said in that video, it holds the large intestine to that back part of the body, keeping it together. And mesentery proper, difference with that is that it holds the small intestine in place, so it's not moving all over the place. So when you think of mesocolon, think of your small intestine, how it's not moving too much. When you think of a mesentery, that's on your colon. Okay, going into more of the serious stuff, into the gland side of things. So we have salivary glands. So a great way to memorize salivary glands. So this portion of the exam would be more on histology. So ways to memorize salivary glands. So here right now we're looking at, in this picture, a submandibular gland of a monkey. So submandibular, it's the area low in the lower part of your mandible for a monkey. So important things to look for in a salivary gland slide is serous cells, mucus cells, and salivary ducts. So purple color and pink. So a great way I try to remember this. So if I was to look at a slide like this and I see a bunch of purple circles with some whitish / pinkish parts in between, and with these little, I call them kiwis, but they're actually called ducts. If there's any of these kiwi duct things, then I know I'm looking at a salivary gland. So serous cells, so things like these, purple, like grapes, are serous cells. Things like things that are pinkish, like these right here, are mucus cells or mucus acini. Acini stands for clump of cells. So in that arrow right there is pointing at those white circles, but actually those circles are actually slightly light pink mucus cells. And the way I remember this is so there's pink bubbles on the grapes. So that means there's mucus producing cells on serous cells. Okay, on to the salivary ducts. So salivary ducts, can't forget, have cuboidal and columnar cells. You can notice by how close the nuclei are, very close together, very cuboidal, very columnar. And of course, when we think of cuboidal and columnar, you think absorption and secretion, hence salivary ducts for salivation secretions. So a bit more on the function side of things, the serous cell secretes salivary amylase. It's an enzyme that breaks down polysaccharide chains of carbohydrates. Mucus cells are pale pink cells secreting mucus to keep the mouth moist and lubricate food as it is chewed. Something interesting about mucus, mucus is very alkaline, which is contrast to stomach acid, which is very acidic. Salivary ducts are pinker, simple cuboidal / columnar cells. Okay. Onto the esophagus. So our esophagus. So our esophagus looks like this. There's a set of layers if you haven't noticed. So a great way I'm going to try and remember when I'm looking at an esophagus slide is look for these little purple purple little folds on the top going up and down like that, as if it's like a gland or something. So that purplish area is known as the epithelium, with a backing of connective tissue known as the lamina propria. And of course we have a muscularis mucosae layer right under that, and a submucosa right under all of that. So this is the esophagus of a human. That's what it would look like. Here's another view of an esophagus at 4 times magnification. Another view of the esophagus at 10 times magnification. So you can really see the epithelium here, the little folds on the top. Another different view in the esophagus right here. You can see some of the folds right there. And it's pretty much all over the place on that picture. Here's another picture of an esophagus. You can see the mucosa on the top, very noticeable, not very purplish. This picture is very bright pinkish. You can see the lamina propria on the backing. You can see the muscularis mucosae there, and mucus acini, or the mucus cells of the esophageal glands. Yep, our esophagus has some mucus cells also. Then there's the submucosa and the muscularis externa, which is not viewable on any of the other slides here. So there's also an inner circular muscle layer and an outer circular muscle layer and an adventitia, but the slides here don't go that far, only up to submucosa. Epithelium. So this is a picture my professor put on the board. We did an experiment with digestive enzymes using lactose and its breakdown from about 15 minutes to half an hour to 45 minutes to until it's about 2 hours. We had to monitor the progress, and what basically happened was lactose was broken down using in 4 tubes. So the first tube was like a control where no enzyme was added for breakdown. The second tube was lactase was added, which produced products such as galactose and glucose. And the third tube had acetic acid added, and that kind of slowed down the reaction. And the fourth tube had sodium bicarbonate added, which sped up the reaction. There was also yeast in all of the tubes. So here's a picture of more of the epithelium. So that's kind of like an esophagus. So far we went over salivary glands, what's important in that, serous cells, mucus cells, and ducts. Then we went into the esophagus, more on the epithelium, mucosa, lamina propria, submucosa, serosa, but we don't really get too far over submucosa. So you can see lamina propria is more on the folded area here. Some things my professor wanted us to look for: purple acini, serous cells. Now we're talking about back on the salivary gland histology slides. White acini, pink mucus cells, and rings of purple nuclei. Those are the with pink cells. Those are ducts. So now we're looking at a stomach. Now we go all the way until the muscularis external layer. So stomach human, we have a mucosa on the top, then a submucosa, then a muscularis externa bottom layer. So the mucosa of the stomach has simple columnar epithelium, which makes sense because there's going to be some secretions going on there. There's some gastric pits that have goblet cells. This is the stomach fundus, or the outer layer of a human. So there's a submucosa and the muscularis right there. So there's the stomach of a human, better labeled: mucosa, submucosa, muscularis externa. So the difference between a stomach histology slide and a esophagus slide is that notice how there's a pink, very pinkish, brightish muscularis externa at the very bottom. That very much stands out. And the submucosa is a mid-layer that's very thin. You can see in both pictures the submucosa. It's another view of the stomach of a human, but magnified by about 40 times. So you can really see in these pictures the simple columnar epithelium surrounding the edges of the mucosa. For secretions, then we have the gastric pits, which are large dips in the edge of the mucosa. And then we have gastric glands, dense purple stains. Gastric glands are usually in the very bottom. Yeah, something like that. So, simple columnar epithelium are surrounding the edge of the mucosa. Gastric pits are the large dips in the edge of the mucosa, and gastric glands are dense purple stains. Gastric glands are involved with having hydrochloric acid and proteolytic enzymes such as pepsin. More on the stomach of a human at 40 times objective lens. The submucosa appears shredded because collagen fiber bundles split during slide preparation. I mean, you can't really see much of a submucosa here. Some more slides of a stomach here. Yep. You can really see the glands and a bit of the epithelium right there. Bit of a shredded top right there, but you can still see the mucosa, lamina propria, submucosa, and the muscularis externa right there. You can really see the simple columnar epithelium on the top on the mucosa in this picture. Another one of the stomach fundus, except this time instead of humans for the monkey. So notice how the mucosa is very much different from a human. The simple columnar cells on the top are very much more pinkish. Submucosa is very much all over the place. Muscularis is not very colorful as a human, very pale colored. Duodenum of a human, muscularis mucosae. The thin layer between the mucosa and the submucosa. So you can really see the muscularis mucosae there. It's very much red. Mucosa is on the very top with some microvilli on the top also. And the submucosa in between and the submucosal glands can also be seen. Muscularis externa at the bottom, and at the very bottom, of course, we have a serosa. This is a duodenum of a human. Another closer view of this human duodenum, you can see the villi, which has simple columnar epithelium. There's also intestinal crypts between the villi. Yeah, intestinal crypts. There's There's also submucosal glands, and the lamina propria inside villi is in between crypts. Lamina propria is inside villi. More on the duodenum of a human. So you can see different versions of a duodenum. You can still see the microvilli there. Muscularis mucosae, a bit more zoomed in over here, times 4 times 10. You can see the microvilli, even closer, 40 times objective. You can really see the simple columnar epithelium there. Small intestine and duodenum of a human, very much red here. Duodenum upper of a monkey, so microvilli again. Notice how the colors are much darker than a human. You can really see the microvilli, everything is very much noticeable for monkeys. Microvilli, submucosa, microvilli mucosa, submucosa, muscularis externa, and submucosa. Closer view of the glands. The ileum. So the ileum, so that's the last part of the small intestine before it reaches the large intestine. So the ileum of a human. So looks very much like our duodenum pictures we've been looking at. It looks very much like our duodenum pictures we've been looking at, with very noticeable villi. Except now for ileum, we need to focus more on the plicae circularis. It's where folds are too large to be villi, but villi is present. Lots of folds here. Onto the large intestine. So large intestine, we have to look out for, once again, simple columnar epithelium. Anything that has secretions really has simple columnar epithelium. The intestinal glands, goblet cells, lamina propria, muscularis, and lymphatic nodule. So the intestinal glands are those circular things right there. Very circular. Next, the lamina propria, that very pinkish lower layer under all the intestinal glands, or the mucosa. Lymphatic nodules are very much dark purplish, not much inside them, very dark purple circles. Submucosa is below the lamina propria layer. Okay. And we have all the way in the bottom, circularis muscle or muscularis externa. So this is the large intestine. Closer views of the large intestine. So what we're looking for here is goblet cells. So we can see the large intestine, much folds. You can see the muscularis externa, very noticeable dark pink color there. So now we're looking for, okay, there's simple columnar epithelium, and there's also goblet cells inside of the intestinal glands. Yeah, they're inside of the intestinal glands, those little circles in them for the large intestine, those are goblet cells. There's also lymphatic nodules here, those dark purple circles right there, with the ones that don't really have goblet cells inside them, those are lymphatic nodules, so they're very close to the lamina propria. Intestinal crypts also exist here in between the glands, I believe. Onto the appendix, so what we're looking for here are simple columnar epithelium, mucosa, submucosa, muscularis layer, goblet cells, and lamina propria. So here we have an appendix. We can really see those, or here, a human appendix. We can see the muscular layer. It's going from bottom to top. So now the mucosa looks like it's on the very bottom. Remember, this is an appendix. It's very much different from the other things we've looked at. That's the one at the top is a muscular layer. Next is the submucosa, then the mucosa. Another picture. Looks like the muscularis externa is much more on the left than this one. Muscularis layers in the back in this one. And submucosa, mucosa. Here's an appendix of a human. More pictures of appendixes. Another picture. You can see probably those white circles are some mucus cells or goblet cells. Could be goblet cells. Salivary glands. Look for mucus, salivary ducts, and serous cells. So you can see it's pointing more on the grapes here. In between there's kiwis or ducts. Mucus cells, very much pinkish in this picture. Salivary gland. Okay, salivary gland. Kiwis look much different there. The esophagus has stratified squamous epithelium. for abrasion. There's muscularis mucosae. You can probably see the muscularis mucosae on the top. And there's lamina propria right under all the mucosa. Then onto the stomach, we have to look for gastric glands, simple columnar epithelium, gastric gland, gastric pits, mucosa, submucosa, muscularis layer. So the mucosa is obviously the one on the top. Submucosa is the right below that mucosa. Then the muscularis layer is the very bottom. Gastric pits probably much closer. We can probably see the gastric pits and the glands. Simple columnar. Duodenum also has simple columnar epithelium. So the esophagus and the stomach and the duodenum, of course, involved in secretions. So simple columnar epithelium. So there's gastric. So the duodenum has gastric glands, villi, which is very noticeable here. You can kind of see the glands too. Lamina propria, lower layer of the mucosa. Submucosa in between right there. And the muscularis layer. Ileum. So for the ileum, we have to look out for plicae circularis. So these things where the folds, and folds in front of them. And there's also a muscularis mucosae layer on the outside, that pinkish layer. Plicae circularis in the middle, muscularis mucosae on the outside for the ileum. Then onto the colon/large intestine, once again looking for lymphatic nodules, goblet cells, lamina propria, submucosa, simple columnar epithelium, intestinal glands, and muscular layer. We can kind of see it right here. The goblet cells inside those intestinal glands. The submucosa, that's the mucosa up there. I think that there's some lymphatic nodules here, not very much purple circle, but of course, since there's a colon, simple columnar epithelium. Intestinal glands, very noticeable with the goblet cells inside them. Muscular layer at the very bottom. And that's it. Okay, on to another thing my anatomy lab professor does is she tests us using figures. So we need to find the spots on the figures. So things we're going to be looking for here are the Brunner's gland or the intestinal gland, the muscle cells of the villus, the longitudinal muscle layer, the longitudinal muscle layer is one at the very bottom. Okay. The lymph follicle or lymph nodule, the muscularis mucosae, cell-rich some of lamina propria, villus, goblet or mucus cells, columnar epithelium of mucosa, opening of Brunner's gland or intestinal glands into the crypt, circular muscle, squamous epithelium of serosa. Those are the ones I have to look for here on my lab practical tomorrow. So yeah, guys, slides really do help. Here's a bit of a video. Okay, that was my friend's video. So a bit of colon pathology. Some diseases of colon can have appendicitis, cecum, the Crohn's disease, enteritis, intestinal disease, tinea, spastic colon, hepatic fixture, adhesion, ulcerative colitis, diverticulitis, diverticulosis, diverticula, flexure, Hofstra area can have bacterial infection in our large intestine. Descending colon cancers obviously could possibly is also an issue. Polyps, sigmoid colon. Polyps can affect the sigmoid colon, and all of this can be seen in this figure. All of these different problems that could occur in a large intestine called colon pathology. Some pictures to help visualize the large intestine and small intestine. Okay, on to the enzyme digestion experiment with lactose. Notice how the test tubes are very different. Test tube number 1 didn't really react. Test tube number 2, there's a bit of reaction. Test tube number 3, there is a reaction, but it's very much slow. Test tube number 4, use the base. Pretty fast reaction, sodium bicarbonate. Then we do some comparison. Test tubes numbers 3 and 4. Number 3 reacts slower, number 4 reacts faster. Acids versus bases. Test tube number 3 with the acid acetic acid reacted slower, and test tube number 4 with the base reacted much faster. Some reactions that were taught in class. Lactose is broken down using lactase to make glucose and galactose, which are both monosaccharides. So you're breaking a disaccharide, lactose, into monosaccharides, glucose and galactose. Glucose can be broken down even more through glycolysis into pyruvate and oxygen, and if it can be broken down even more to acetyl-CoA, which undergoes citric acid cycle and electron transport system, producing 32 ATP and H2O. Pyruvate, on the other hand, besides oxygen, pyruvate can turn into ethyl alcohol and carbon dioxide, C2H5OH. And that's it, guys. Thanks for